Hello everybody. I am Dr. Farhan Zamir, an adjunct professor and academic specialist at Biotechnica, Bangalore. For today, we are trying to discuss a very important topic that is on demand. On Biotechnica office, we were able to get a lot of queries on the application of bioinformatics, looking into our previous videos, and there were few major queries asking for certain specific applications of bioinformatics in cancer research. So this video is dedicated for all cancer researchers, wherein they want to implement bioinformatics in their cancer research. So let's try to understand, let's dwell in. So for today, we will try to discuss on a very important and advanced topic, which is called as application of bioinformatics in cancer research. So this was on huge demand, and hence they were asking for help in bioinformatics, especially in oncology. We thought we will try to share this particular knowledge. So let's start. Now, to start our discussion, I have taken up an example of colon cancer. So please remember, whenever you talk about cancer, you need to first understand the etiology and the various stages of cancer progression. So in our discussion for today, I have taken up colon cancer as one such example. So in colon cancer, we will look upon how exactly colon cancer progresses. So you have the first stage wherein just a few cells which are abnormal, which are fickle in nature, would start replicating in an abrupt phase, wherein without the checkpoints, that is breaking up all the checkpoints of your cell cycle, this would start their progression. Once the cancer cells progress, they will lead into a series of stages, which is stage 2, stage 3, stage 4 and stage 5. In most of the conditions, especially with cancer, colon cancer or colorectal cancer, this has been only detected at the stage 3 or at the stage 4. So by then it would be too late. So there are various things where we can look cancer research in the eyes of bioinformatics. Now the first thing is, am I trying to design a drug for colon cancer, or am I using this particular strategy of bioinformatics so that I can detect a diagnostic marker, or can I design something which would be a therapeutic target or a preventive target? So all this, so first, before you actually look into the various components of bioinformatics and their specific tools, you need to first define what exactly is your hypothesis and where exactly you are trying to knit the story. So moving forward, how can I use bioinformatics? Now the first tool is to understand which type of cancer I need to study. So for this, you have a beautiful database which has been a dynamic database generated by and maintained by WHO, that is World Health Organization, which will tell you the entire statistics of the mortality and morbidity of a particular country. So it is not on a particular race or a particular population, it is on a country wise. You have huge amount of data which is dynamic data, and I have taken up an example of 2017 data, and on a 2017 data from a Japanese population, what exactly was the number of deaths, how many people died just because of cancer, in that how many were segregated as males and females, how many of these cancer patients actually suffered with cardiovascular disorder, and then what is the death ratio in terms of their age, especially from congenital disorders to old age disorders. All this information is available on a dynamic database maintained by WHO. So please, I want you people to refer this so that you can have the mortality and morbidity ratio. Now this will tell you the importance of your study. Why should I study colon cancer, or why should I study a typical type of cancer? Now, I took up a case of Japan with a particular logic, so that if I give you a logic, you will be able to extrapolate the same logic to your other research. So the logic is, Japanese, they eat hot food. So when they consume hot food, when I say hot, it is really hot. So the minimum temperature of this food should be around 60 to 70 degree. Now, when it is 60 to 70 degree, when they're consuming it, it is obvious that their esophageal and in the worst scenario, the stomach and there are the digestive tract problems which can arise. So here with a Japanese database, I could get excited to study what is the incidence of esophageal cancer in Japanese population. So I have knitted my story and now I know the purpose of why am I studying this kind of a disorder. So, first define your purpose. So once you define the purpose, there is a lot of things what you can do in terms of cancer research. The first thing is, and the booming component is, you can look into the cell biology component. And trust me, dear friends, there are a lot of data which is already available on cell biology of cancer. And if you want to extrapolate that into much deeper sense, you can understand the fundamentals of cancer. Now, understanding the fundamentals of cancer can be extrapolated into beautiful branch of cancer or oncology, which is called as regenerative cancer. Now, in regenerative cancer, you are trying to either manage or inhibit or actually nullify the entire tumor process by various kinds of approaches. So this particular diagram actually shows the various approaches in which you can, look forward. Now, let me quote few examples and then move forward. Now, if I want to study the microbiome and the inflammatory mechanism related to colon cancer as our example, or am I interested in looking into the cell growth, differentiation, or something related to stem cells, or tissue regeneration pattern, or can I look into an integrative signaling mechanism, because cancer is all about signaling. So when I talk about integrative signal mechanism, it is not just I'm looking at the colon cancer per se, but however, what exactly the colon cancer undergoing metastasis can lead into a different type of secondary cancers in the other component of your body. Or I can also look into what is the drug resistance and what exactly I can do in terms of targeted therapy. Or can I look into the controlling up of the cancer cell so that it does not multiply? So I am trying to control the cell cycle rather than the checkpoints which are open. Can I somehow put up a COP or a ligand molecule which will actually revert back the checkpoint? So the cyclin dependent kinases, normally they get dysregulated, and that dysregulation I can study in terms of gene regulations. Either a gene could be upregulated or downregulated. So the bottom line of this particular slide is to make you understand that once you define what type of cancer you want to study, in what population you want to study, you need to first define an approach. So your approach should be of an inverted pyramid mode, wherein to understand this, maybe I want to study something called as exosomes, because exosomal research is now into its peak and it's into boom. So, maybe I want to take up exosome, and in exosome, I want to study the targeted specific cells wherein how exactly a drug could actually affect a particular target. Or what happens if there is exosomal clearance, or what happens to a particular tissue or a cell. And then very importantly, I can also design certain biomarkers which can actually help me in imaging and tracking. So or at a biochemical level, I can look into its post-translation modification, or also into its epigenetic components, wherein I can look into my miRNA, I can look into HDACs, etc., etc. So the moral of all these particular slides until now is define your story. So you need to know what exactly is the target and what is that you are looking into the system, and bioinformatics would be a tool to work on it. Moving on forward, you need to also define the tissue. Here, the tissue specific target becomes very, very important so that we can we can understand it, in a much better way. Example, am I looking into the, brain biomarker, or a heart biomarker, or a kidney biomarker, or a gut biomarker, muscle biomarker, blood biomarker? So defining a particular tissue becomes very, very important, because cancer could be in any of the tissue. Cancer can happen or can spread in terms of metastasis to any of the tissue. So understanding, knitting the entire story becomes a primordial factor for a cancer researcher. Now, once I have identified the tissue, so the next thing is, in a tissue, which is the biomarker I am interested with. So I have given up a series of biomarkers and their tissues in which these particular biomarkers are normally expressed. Say for example, if I take up a biomarker such as esophagus. So in the esophagus, a biomarker called as CEA is a biomarker which is normally expressed. Or if I take up a uterus, or if I'm studying a uterine cancer, then you have SCC and then CEA and other biomarker. Then if I want to study a prostate biomarker, then it is PSA as an other biomarker. Or if I want to study breast cancer, maybe an ER, that is estrogen, or HER2 or PR could be an other biomarkers which can be involved. Now, my intention of the study is I have identified what exactly is the tissue biomarker. Now, the interesting component is how is this esophageal biomarker connected to a uterine biomarker, or how is this uterine biomarker connected to breast cancer biomarker. So I need to understand, and it is impossible for me to do a manual curation and try to understand this biomarker. And here is the point wherein bioinformatics comes into your rescue. So what I do is, I have selected, to make you understand this component, I have selected few biomarkers from our previous list. That is, I have TPA, I have CEA, HER2, BAF, ER, that is estrogen receptor, BRAF, then you have certain bone receptors, then you have thyroid receptor which is called as thyroglobulin, etc., etc. Now, using bioinformatics, very importantly, string database, which is absolutely for free to try. Okay, so using string database, I am taking this particular combination of biomarkers and trying to look into the functional network. So when you look into the functional network, you see that in my story, I believed that CAA is a major biomarker which is actually involved in the process. But now you are very much evident that, CAE is not involved in the pathway at all. In the pathway, what we have constructed, it is only this component which are involved, and the upregulation, downregulation, or the co-expression only takes place with this, and CEA is not at all involved. So this gives me an idea, the string database gives me an idea to understand the functional analysis and development of a network to understand what exactly is the expression in terms of its data curation and in terms of its experimental studies. So this has now been a great impact on designing pathways. So once I understand the string database and the biomarkers and the tissue specific biomarkers, then the next step is to develop the pathway. Now, until then, I designed a very simple network, and then you would have thought that, oh, sir has taken only 5 or 6 biomarkers and the story is very simple. But that's not the case, my friend. So when you look into the story, this is the complex. So here, each biomarker, so we were talking about RAF, which is one of the major biomarker, BAF, another biomarker, and CEA, which is another biomarker. When I look into a bigger picture, so what we understand is HER2 is involved with CEA with only one string. So you can see there's a beautiful one string. So this means to say, if I am able to upregulate or downregulate HER2, then this has a direct effect or an indirect effect on CEA. And we also looked upon thyroglobulin, and its interaction could be also looked into. So the important component is this will give you an idea of how exactly the components or the biomarkers or the biotargets from another tissue or the same tissue are interlinked with each other, and this gives a very important baseline for me to do any kind of a wet lab study, especially cell line studies. Now, this database is not just trying to give you a network which will talk about the expression and the other things, but this will also make you understand the co-expression between the genes and the proteins. So wherever you see this component, okay, on your x-axis and y-axis, you have this particular biomarkers, and how are these biomarkers interconnected, and expression of one could lead into the downregulation of the other, and the higher the expression, lower would be in a downregulation of another biomarker. So there is an direct relation or there is an indirect relation between the biomarkers. So one promoter could be an inhibitor for something else, and other inhibitor could be a promoter for something else, and understanding this entire scenario becomes very, very important, and it is through this particular string database analysis we can look into the co-expression of the target selected. To study this in a much better way, and the introduction of a chemical molecule, because finally I want to treat this cancer. To understand or for treating this cancer, I need to have a chemical molecule, which we call it as a ligand molecule. Now this ligand molecule could be from a herbal source, it could be from a phytochemical source, or it could be from my chemistry laboratory, so it could be a synthesized chemical. So, or I have the parental molecule and I can lead into the changes in the functional group. As you see in the figure, this is vincristine, a known anti-malarial drug, wherein you can work for various kinds of systems, and this is many a times it has been also looked upon as a promising molecule for anti-cancer agent also. So here what I have done is, this is the entire structure of vincristine, but I have introduced OH group wherein I am trying to derivatize the molecule so that I don't know the influence of OH group can actually increase the rate of the reaction or decrease the rate of the reaction, and this combination of changing of the functional group is called as derivatization, which plays a very, very important role in a new emerging field which is called as Cheminformatics. Once I have understood the nature of the molecule, which is called a ligand, now we need to see what is the vincristine doing with a particular cancer pathway. Now, vincristine can actually arrest the mitotic cell, or it can also lead into the mitotic death, or it can actually hamper the cell cycle. Now, in cell cycle, you have various phases like G1 phase, S phase, G2 phase, and M phase, which is mitotic phase. And what vincristine is actually trying to do is it can actually block the G1 phase with the microtubular sensitivity checkpoint being blocked, or it can also lead into the mitotic arrest and mitotic death. So this is how there is a story which has been knitted around vincristine and its control over the cancer pathway. Now, to dwell upon more, this can also have a profound effect in looking into its microtubular interaction. What exactly is vincristine doing on a cancerous cell and its biomolecular mechanisms like DNA, RNA interaction, and we can also see what exactly the interaction of the vincristine on a mitochondrial biomarker. Okay, and especially which can lead into the various dysbiosis which can happen during a cancer environment, and very importantly how exactly it can activate or deactivate certain components of the immune system. So all this could be studied using bioinformatics. Now, a very important component wherein a lot of people are trying to look upon is how exactly a ligand molecule is interacting with a target molecule. Now, target molecule could be any molecule such as a receptor, could be a protein, could be a nucleic acid, could be RNA, could be DNA, could be a receptor, whatever. But here, the important component what we are trying to understand is how exactly a ligand molecule can interact with your protein. Now, when I say interact with the protein, if this is a protein, remember the ligand molecule does not go and hug the entire protein. The interaction is at a very, very particular site, and this particular site on the target is called as an active site. So in this figure, very clearly you are able to identify the ligand molecule, and this ligand molecule goes and binds to a particular site at the target, and this is how we call it as QSAR studies, the quantitative structure activity relationship studies, could be understood using bioinformatics. Now, it is not just one ligand molecule going and binding to the receptor. You can also see the chemical interactions like hydrophobicity. You can look into the negative or positive electrostatic interactions. You can also look into the van der Waals forces, and you can look into the hydrogen bond formation, what is the bond length, what is the bond energy, and very importantly, ultimately defining the atomic contact energy value, what we call it as glider's value or ACE value. Once I understand all this background information, to extrapolate, I want to look into the sub-molecular levels. So to look into the sub-molecular level, maybe I will have two groups. One group is the group which is actually suffering with cancer, okay, and the other group is the patient who is suffering with cancer, but it has been treated with my tested ligand. So here in the figure, with using a program like R, in cancer research, I can design something which is called as PCOA analysis curves, that is principal coordinate analysis curves, which will actually define the cluster among the demographic patients what I have studied. It will help me in designing clusters of a particular group, which will define whether my treatment is effective or not. Now, to understand that, I can also go into the set of biomarkers which are actually responding, and then pick specific set of biomarkers or specific set of biomarkers which will tell me that these are the indicators of cancer or these are the indicators for diagnostic markers. So this Venn diagram will actually define the similarities and the uniqueness among the two groups. Now, with this as a data, using R programming or any programming from Python or from BioJava, BioPerl, you are able to create this heat maps. Now, this heat maps will tell you the expression pattern, whether the set of genes are upregulated or the set of genes are downregulated. This could be studied using these particular heat map. So in the cancer tissue, the regulation component could be studied using bioinformatics. Now, very importantly, to mimic what is happening in the cancer environment, you can also look into the molecular stimulation and dynamics data. So in a real time, what is happening to a cell in a lipophilic or lipophobic environment could be addressed using molecular stimulation studies and dynamics. This will ultimately help us in identification of the target, enrichment of my pathway, will help in understanding the gene clinical feature association, the gene module clinical feature association, and in turn at the final component, which will help me in patient stratification. So all this is a pipeline which is normally used in cancer research. Now, with all the information, we are able to also look into the KEGG database, wherein the entire metabolic network and the microenvironment which is happening into a cancer cell could be studied using KEGG database, that is Creative Encyclopedia for Genes and Genomes, which will help me in designing pathways. So once I define pathways, I can look into few newer biomarkers, maybe not directly involved into the cancer environment, but however, which can influence the growth or progression of cancer. These are called as inflammatory biomarkers, or there are immune subsets. Say for example, I take sub T cell subset, wherein I can take up Th1, Th2, Th17 or T regulatory cells, which are actually influencing the microenvironment of cancer. So that's why in the beginning of my video, I just told you that we need to first properly knit a story so that the way we progress can be enormous. We can do a lot of wonders once we understand the purpose. Now, once we understand this, we can also look into the microenvironment and how exactly the colon cancer is being regulated and what are the other various components of cells which are actually influencing the cancer microenvironment and its toxicity. Now, a huge amount of research is going on in colon cancer, wherein people are trying to focus on gut microbiome. Now, rather than giving a specialized drug which has a lot of side effects, say for example, people with colon cancer, they normally go for radiation therapy, chemotherapy, wherein the patient is simply not able to sustain after third sitting or fourth sitting. So the patient collapses. So can we design something which is actually a component of his body? So if we are able to regulate that particular component, somehow I am able to at least not treat, at least manage the disorder. And gut microbiome gives a huge promising factor, wherein huge amount of research is actually being worked on gut microbiome in treating cancers and various such kind of disorders. So to understand gut microbiota, again there are a lot of analysis with the 16 years rRNA data which has been available in terms of bioinformatics resources. Time and Mother are the two major freely available tools which are being used in analysis of gut microbiota in bioinformatics. So what is the intention? The intention is from the traditional medicinal component, wherein we have a patient, we give a particular drug, we look into trial and error method. If the disease has been treated, well and good, or if the patient is lucky, or if he has been blessed, well and good, otherwise the patient collapses. Now, there is a lot of, luck factor which has been involved. But however, with the advent of bioinformatics, what people are trying to discover or invent is something which is called as precision medicine. So this means to say, rather than me having a lot of side effects, can I do something wherein the drug actually goes and binds to the target, has a profound effect, so that the patient recovery is almost near to 100%. So this is what we are trying to design, which is called as precision medicine. And in cancer research, precision medicine is the technology for future. So anybody who is an oncologist, anybody who is a cancer researcher, we urge you that with the current lifestyle, I think there would be a huge population of people who are suffering with cancer in the near future. It is you, the hope, wherein doing all these kinds of advent of technology with biology, understanding the components in a much better way, can lead into precision medicine or something which is called as personalized medicine. We look forward from our budding scientists for these kind of inventions and creativity. So this was a gist of how exactly you can use applications of bioinformatics in cancer research. So if you have any suggestions, please do not forget to write to us. And also if you would like to look into these kind of videos which are much more informative, we are very happy. And please let us know which would be your research of interest, and we will try to we will do a small research, and we will try to design videos for your research. Our mantra is your success is our priority. Thank you very much. Thank you for your time.